Could you imagine a future where genetic disorders become a thing of the past? This future may not be as far off as you might think. In the realm of medicine and biotechnology, we stand on the precipice of a new era, one where we have the ability to literally rewrite the code of life. The key to this future: gene editing. Gene editing is the process of making precise and targeted modifications to the DNA of living organisms. This could mean correcting genetic defects, treating and preventing the spread of diseases, or improving crop yields in agriculture. Among the tools ushering in this new era, one stands out for its precision and ease of use: CRISPR-Cas9. This revolutionary tool, derived from a naturally occurring genome editing system in bacteria, is changing the game in genetic engineering, opening up possibilities that were once only the stuff of science fiction. Welcome to the fascinating world of CRISPR-Cas9, where science fiction is becoming scientific fact. You may ask, what exactly is this CRISPR-Cas9? Well, imagine you're working on a document in a word processor. You spot a typo and you use your keyboard to correct it. Now imagine doing the same thing, but instead of a document, you're working on the genetic code that makes up every living organism. That, in essence, is what CRISPR-Cas9 allows us to do. CRISPR-Cas9 is a revolutionary gene editing tool, and to truly understand it, we must delve into the heart of our cells. Within each cell, we find our DNA, the blueprint of life. It's a long, complex string of molecules that tells our cells what to do and when to do it. Sometimes, however, this string may contain errors, genetic mutations that could cause diseases. This is where CRISPR-Cas9 comes in. CRISPR stands for clustered regularly interspaced short palindromic repeats. These are sequences of DNA that bacteria use as a kind of immune system, allowing them to recognize and defend against viruses. Cas9, on the other hand, is an enzyme that acts like a pair of molecular scissors, capable of cutting strands of DNA at specific points. Together, they form a system that can locate specific sequences of DNA within the vast complexity of a genome and then cut and replace them. It's like using the find and replace function in a word processor, but for our genes. This revolutionary tool allows scientists to target and edit genes with an unprecedented level of precision and efficiency. It opens up a world of possibilities, from curing genetic diseases to improving crop yields and even potentially bringing extinct species back to life. But it's not just about editing genes. It's about understanding the very fabric of life itself. By giving us the power to manipulate our genetic code, CRISPR-Cas9 grants us a deeper insight into the fundamental mechanics of biology. Thus, CRISPR-Cas9 is our precise and efficient tool for editing genes. And with this tool, we're on the brink of a new era in science and medicine, one where we're not just reading the book of life, but writing and editing it too. But why does this matter? What does this mean for us and our future? Imagine a world where genetic diseases are a thing of the past. Imagine a future where our crops are more resilient, more nutritious, and able to grow in harsh climates. Imagine a world where we could combat climate change not just by reducing our carbon footprint, but by actively altering the genetic makeup of organisms to sequester carbon dioxide. It sounds like the plot of a science fiction novel, doesn't it? Yet with CRISPR-Cas9, this could be our reality. CRISPR-Cas9 holds the potential to revolutionize medicine as we know it. It opens the door to a future where genetic diseases like cystic fibrosis, muscular dystrophy, or Huntington's could be cured before a person is even born. By precisely editing the DNA within our cells, we can replace faulty genes with healthy ones, eliminating the root cause of many inherited diseases. Agriculture and food production stand to benefit immensely as well. With CRISPR-Cas9, we can make our crops more resilient, enabling them to withstand pests and harsh weather conditions. We can engineer plants to be more nutritious, addressing malnutrition and food insecurity in many parts of the world. And then there's climate change. It's the defining challenge of our time, and CRISPR-Cas9 could be part of the solution. By harnessing the power of this technology, we could potentially engineer organisms to capture and store carbon dioxide, helping to reduce the levels of this greenhouse gas in our atmosphere. But it doesn't stop there. The potential applications of CRISPR-Cas9 are as vast as our imaginations. It holds the promise of a future where we can not only understand but also manipulate the very building blocks of life. The potential of CRISPR-Cas9 is vast and transformative, truly a revolution in the making. However, with great power comes great responsibility. As we delve deeper into the potential of CRISPR-Cas9, it's important to acknowledge the ethical and safety considerations that come along for the ride. One cannot overstate the risk of unintended mutations. This technology, while powerful, isn't perfect. An unintended cut here, an unexpected edit there, could lead to unforeseen consequences, not just for the individual but potentially for future generations. Then, there are the ethical implications of altering human genes. The line between therapeutic use and enhancement can blur quickly. Who gets to decide what constitutes an improvement? And what about consent? Not just from the individual but from their future offspring who will also carry these edits. These are questions that don't have easy answers. While CRISPR-Cas9 holds immense promise, it's crucial that we navigate this new frontier with caution and wisdom. As we stand at the precipice of this revolution, we must ensure that our actions are guided by a commitment to do no harm. So, what does the future hold for CRISPR-Cas9? As we stand on the brink of a new era, the revolutionary potential of CRISPR-Cas9 is undeniable. This groundbreaking technology is poised to redefine our understanding of biology and medicine. Imagine a world where genetic diseases are a thing of the past, where crops are engineered to be more nutritious and resilient, where we can literally rewrite the code of life. But as we step into this brave new world, we must tread carefully. The power to alter the genetic code comes with enormous responsibility. Ethical and safety considerations must be at the forefront of our minds, guiding us as we harness the power of CRISPR-Cas9. In the hands of responsible scientists, CRISPR-Cas9 could redefine the boundaries of what we thought was possible. The future is not just near, it is here. Let's embrace it with open minds, cautious optimism, and a deep respect for the power we now wield.